So since there is a question about HDL and LDL, so we'll just look at that slide once more and then we'll go to today's class. So yesterday we were discussing about the lipoproteins and their involvement in transporting triacylglycerols and cholesterol. Okay, so primarily we looked at these two lipids. So the question is, let me read the question once more. Now, I didn't get the concept of HDL and LDL with respect to having high cholesterol levels and possibly having high risk of getting the disease. Okay. So HDL, LDL and their levels and how that connects to cholesterol levels. So that's what we are going to focus now. So the first point is we start with these two, chylomicron and VLDL. So chylomicrons transport dietary lipids to other tissues. Okay, primarily to, it carries in the blood and that activates a lipase. And then the free fatty acids produced by these lipases are taken by the tissues either for beta oxidation, like for example muscles, or for storage by again converting into triacylglycerol in adipose tissues. Okay, and after the cargo delivery, the remnants of chylomicrons go back to liver. So this is one. And second, when you have plenty of carbohydrate available, like carbohydrate rich diet, so then the main dietary material here is carbohydrate. Okay, so they are not carried by chylomicrons, so they are by glucose transporter and so on. So we didn't get into that, but anyway, that excess carbohydrates in liver will be converted into triacylglycerols. And from other sources, for example, amino acids after transamination, the carbon skeleton will be again converted to acetyl-CoA and used for making triacylglycerols and cholesterol. And these are, like, instead of intestine, here liver being the source of the lipid, they are packaged in VLDL. Okay, the primary differences are the apolipoproteins that are present, and they act as signals which cells are going to take up and what cargo they carry. So those are identified by the lipoproteins present in them. And VLDLs again circulate to other tissues via blood, okay, through these capillaries. They again would do the same thing that happened to the dietary lipids. And from the VLDL, when triacylglycerols are removed, then their density slightly increases. From very low density lipoprotein, they become LDL. Okay, so the LDL is essentially rich in cholesterol, and that should be taken up and used for storage or for energy production by other tissues, extrahepatic tissues. Okay, so their level in the higher level in the blood means higher levels of cholesterol that actually needs to be used. And if that is not used, then that is what deposits on the blood capillaries, blood vessels, leading to a disease called atherosclerosis. Okay, so I think that spelling is coming in the next slide. If not, I'll write it at that point. Okay, so this is what leads to cholesterol deposition, not just cholesterol, even other lipids deposition on blood walls inside. Okay, therefore the lumen gets narrowed or constricted, which increases blood pressure because heart has to pump the blood now through a narrower tubes, so the pressure increases. And if these plaques size really increases and reduces the capillary further, then the blood flow gets obstructed and then you get a cardiac arrest or heart attack. Okay, so that is when the doctors say this artery is blocked and then we are clearing it up. They find that by using a process called angiogram. So that helps them to find where the block is. And then they try to remove it up using angioplasty or surgery, mostly angioplasty. So this is basically a balloon that goes and blows up the obstructing material. So this is the connection of LDL to the disease. Okay, LDL, higher level of LDL indicates higher level of cholesterol in the blood that has no place there. It should actually be taken up by the tissues and used up. So we will see that a disease condition we will discuss in today's class. So that's the LDL. And the difference between LDL and HDL is again apolipoproteins. So the HDLs actually, on the other hand, take up any extra cholesterol anywhere and they take it to the liver for metabolism. Okay, HDL level high meaning you have that HDL particles plenty, so any extra cholesterol can be easily ferried to the liver. So high HDL means high availability of vehicle to carry off the any excess cholesterol that may be there. LDL means you have excess cholesterol loaded, and that is why you have LDL level being high. So a healthy person should have less LDL and more HDL. And there are conditions in which these levels are affected, and that we will discuss in today's class. Dhruv, did this answer your question?
Yes, sir. But
Okay.
I had one more doubt.
Yeah, go ahead.
Why doesn't the LDL combine with the HDL, like all of them? Why does it not incorporated with the HDL so that it gets again retransformed?
Okay. So
Yeah.
these different apoproteins have different functions. Okay, so they have different receptors, and then therefore they have different functions. So HDL on the surface can combine cholesterol and triacylglycerol transported by specific transporters present on the cell membranes. Okay, and LDLs don't do that. On the other hand, LDLs are actually taken up by the cells by the binding of the apolipoprotein on LDL by the LDL receptors on the extrahepatic tissues. So that's going to be a main discussion today. So we'll have a dedicated slide for that. So it is basically the specificity and therefore their functional, delegation. So they have different functions to do. HDL's job is to pick up cholesterol and transport to liver. LDL essentially, after the triacylglycerol is taken away from VLDL, whatever remaining that is primarily cholesterol enriched version is what is LDL, and that is to be taken up by the tissues. So they do different jobs, and that is primarily coming from the different apolipoproteins.
Okay,
so it's basically functional separation. Okay, so today we are going to focus on cholesterol biosynthesis and a little bit discussion on disease condition, and then what are the other molecules synthesized from cholesterol, and then we will begin our discussion on hormonal control of metabolism. So for those of you who are interested in metabolism related diseases, that is actually an entirely separate course. It's there are books, diseases of metabolism, of the size as big as Campbell's biology book. Okay, dedicated only to those, because in each one of the pathways you can have defective enzymes, and therefore different metabolites accumulate, and each metabolite accumulation or the deficiency of the product causes different diseases. So we did see some when we were discussing about lipids in the molecules of life section. So today we will see one about LDL, what are the problems there. So before that, let us begin on the cholesterol biosynthesis regulation. So as you saw yesterday, cholesterol biosynthesis is really a complex synthetic process. Okay, so we skipped the last 20 steps, simply saying that methyl group shuffling and removal of methyl groups and shuffling of the double bonds and so on. Like that, we kind of glossed over it. But whatever we learned itself is complex enough. Formation of mevalonate, formation of this IPP and DPP, and then they joining together to form squalene, and then squalene cyclizing to form the steroid ring. So that itself is complex enough. So you don't want to do all this synthesis, expending a lot of NADPH and ATP unnecessarily. So only when cholesterol is required as a precursor or cholesterol is required for new membrane synthesis do you want to make cholesterol. So cholesterol biosynthesis is regulated at a couple of levels, and both of that we will briefly touch upon. And this was worked out by the same two people whom we discussed yesterday, their work about LDL uptake, LDL endocytosis. We saw, Brown and Goldstein. So they were the ones who identified these SREBPs and SCAP and so on. So we'll briefly go through that. So these are two proteins, sterol regulatory element binding protein, SREBP. Okay, so this is the main regulatory molecule. So it has an integral membrane domain, and that is anchored on the endoplasmic reticulum. Okay, so this green line that you see here. So its N-terminal part, which is not on part of the membrane, this is what is the regulatory domain. And this is in complex with another protein called SREBP cleavage activating protein, or SCAP. When these two together are anchored on the endoplasmic reticulum, they are inactive. Okay. And this SCAP is the one that senses cholesterol. So, sense cholesterol. So it binds cholesterol essentially. So when you have cholesterol, then when SCAP is bound with the cholesterol, when you have high cholesterol in the cytoplasm, then this is going to be bound with cholesterol. And that leads to a conformation that keeps them on the endoplasmic reticulum, presumably involving another protein that probably retains these two on the endoplasmic reticulum. Okay. But on the other hand, if the cholesterol level is low and the SCAP is unbound with the cholesterol, then that releases these two from being on the endoplasmic reticulum and they migrate to Golgi. Okay. So on Golgi, this SREBP undergoes two proteolytic cleavages shown by these arrows, releasing the N-terminal peptide, that is what is biologically active. And this translocates into the nucleus and binds to transcription factors, and this directly binds to the DNA where you have these sterol regulatory element. These are specific DNA sequences upstream of the coding sequence of the cholesterol biosynthetic enzymes. So essentially promoter region of those genes. It binds there and activates transcription of those genes, and ultimately by translation you are going to have the biosynthetic enzymes that then catalyze the biosynthesis of cholesterol. So when you have less cholesterol, then cholesterol biosynthesis is promoted by activating transcription of the genes that encode the enzymes involved in cholesterol biosynthesis. So this is how they regulate. So I told you at the beginning, when you have plenty of cholesterol, then this shuts off. Okay, SCAP binds to it and it is retained on the ER. And as long as they are on ER, they don't undergo, SREBP does not undergo proteolytic cleavage. And this pathway is very fine controlled because this cleaved N-terminal active version in the nucleus is very short-lived. It continuously gets proteolytically cleaved. So only as long as SCAP is not bound with cholesterol, then only you have this N-terminal portion available. And this N-terminal portion should be continuously made. This process, these two arrows must continuously be happening. Then only the gene transcription will be going on, because this molecule gets constantly degraded. Okay, so that is how this is regulated. So this is one mechanism of regulation of cholesterol biosynthesis. So the other one is somewhat familiar to us. So where you have covalent modification by phosphorylation. Okay, so the phosphorylated form is inactive and the dephosphorylated form is active, just like we saw acetyl-CoA carboxylase, okay, for the fatty acid biosynthesis. So the enzyme that is subject to such a regulation is HMG-CoA reductase. So yesterday I told you this is the one that catalyzes the committed step in cholesterol biosynthesis, in general steroid biosynthesis, that is the formation of mevalonate. Okay, so this enzyme is dephosphorylated when you have insulin signal. So insulin will be high when you have glucose high. Okay, so when you have high level of glucose, you want to take up the glucose and convert into storage version, that is cholesterol, triacylglycerol, etc. So insulin, by stimulating AC carboxylase, acetyl-CoA carboxylase, is going to stimulate free fatty acid biosynthesis and ultimately triacylglycerol. In addition, by stimulating HMG-CoA reductase, by dephosphorylating this enzyme, it stimulates cholesterol biosynthesis as well. So essentially, glucose is being taken up and anabolic reactions of these biosynthesis of lipids are stimulated. Okay. On the other hand, if glucose is not available, then you want to mobilize lipids for energy sources and producing the precursors for gluconeogenesis and ketone bodies in the blood. And that is done by blocking the biosynthesis by glucagon. Okay, so the same thing that would block the melanyl-CoA synthetase in free fatty acid synthesis. Okay, so this will, via cyclic AMP and protein kinase A, would end up phosphorylated this enzyme, and that inactivates it. So this is how phosphorylation, dephosphorylation regulates HMG-CoA, and this is integrated to hormone signals again. Okay, and then we know from mevalonate, multiple steps you go to cholesterol, and cholesterol itself has a feedback kind of regulation. One, it suppresses LDL uptake, because LDL is the one that is going to bring cholesterol, and when you have a lot of cholesterol already in the cytoplasm, you don't want to take more LDL into the cell, and therefore the LDL proteins, apolipoprotein transcription, those gene transcription is inhibited by cholesterol. Okay, so that is one role it does, and it also stimulates proteolysis, by an unknown protein, the degradation of HMG-CoA reductase. A third thing cholesterol does is it stimulates this acylating enzyme, this acyl-CoA acyltransferase, which adds to the hydroxyl group of cholesterol. It adds the fatty acid acyl moiety, making cholesterol ester, and that is storage form. This is strongly hydrophobic, which is inside the chylomicron that we saw yesterday. So these are the ways in which the cholesterol biosynthesis is regulated. Essentially, you balance the synthesis with the dietary uptake. When you have plenty of dietary cholesterol available, then you don't synthesize, you turn it off. And instead you take it up and then convert into cholesterol ester and store it. So these are the two important ways in which cholesterol biosynthesis is regulated. So now let us see what are the different, okay, so I told you we will discuss a disease. So in all these discussions, you definitely heard me say several times these apolipoproteins, and these apolipoproteins being specific, they are characteristic for chylomicrons, LDL, VLDL, and HDL. Okay, so when you have deficiencies with them, then you can have problems. And another thing is, yesterday we saw a cartoon, probably the last slide, where I showed you that this LDL is taken up by endocytosis, and for that the apolipoprotein of LDL binds to specific receptors on the cell surface, and binding of the LDL to its receptor leads to endocytosis, and the LDL gets taken into the cytoplasm in the form of endosomes which fuse to lysosome. So now if you have defects in LDL receptor, like for example due to a genetic condition, like you have a mutation, as a result the LDL receptor, the protein produced from such a defective gene, probably is defective in binding LDL. And there is a known such disease condition, genetically inherited disease, and that is called familial hypercholesterolemia. Okay, familial means usually refers to runs in the family, meaning genetically inherited disease. In other words, inborn error, okay, meaning the defect is in the gene. And when you have a defect in LDL receptor and where LDL is not taken up by the cells, then LDL accumulates in the blood, and you have then cholesterol also, in the blood. So when LDL is not taken up, then what will happen? Two things. One, dietary cholesterol accumulates in the blood in the form of LDL. And second, because LDL is not coming into the cytoplasm, this cartoon here, there is no LDL coming in, and therefore the assumption by the cell will be, oh, we need to make cholesterol. There is not enough cholesterol. So the cholesterol biosynthesis gets stimulated, and then you make cholesterol in the cytoplasm as well. So due to this, you have really excess cholesterol, and that's why it is called hypercholesterolemia. This emia sound usually refers to in blood. Something, hypoglycemia, meaning glucose level in blood is low. Anemia, iron is less in blood. Ischemia, low oxygen in blood. So this emia refers to something's level in blood. That's what it means. So here hypercholesterolemia means cholesterol level being very high in blood. That's what this term means. And in such a situation, what you want to do is you want to intervene therapeutically, blocking this enzyme. Therefore, you don't make cholesterol. Okay. And here are those inhibitors. So the common inhibitor were originally found in fungus, and so they are the lovastatin and compactin found in fungi, and they are used to treat. Now we have many synthetic versions as well. So the important point is shown here in the red color here. So this part of this molecule, okay, so when it is R2, it is compacting. So the basic structure remains the same, and by varying these two R2 groups, you have different statins. So these are the statins that the cardiologist prescribes to patients having higher cholesterol or higher LDL levels. Okay. And taking statins have been shown to have very little side effects, and a lot of people having high cholesterol level take this drug on a daily basis forever. And there was a finding that while these drugs reduce the cholesterol level, triacylglycerol levels, and make your blood biochemical parameters really look textbook perfect, they don't necessarily reduce the plaque formation. And people didn't know why is that, but then now we know the reason is these patients are diagnosed very late. Okay, so their arteries are already loaded with plugged with lipids, and it's already narrow. So now reducing cholesterol biosynthesis only will have a marginal effect, not a substantial effect. So currently the thinking in the field is people prone to such diseases must be taking these statins very early in life. Like for example, these are not prescriptions, okay? I am talking about discussions in the research literature. That probably these people should take drugs starting in their mid-20s if they really want to have benefit of these statins. So you look at the structural similarity here. So these molecules, as we learned in our enzyme inhibition, they function as competitive inhibitors, meaning they compete with the mevalonate. Okay, and so that is how they block this HMG-CoA reductase. All right. So this is the action of statins. So statins essentially block the biosynthesis of steroids, and the condition in which steroid accumulation can happen is when LDL is not internalized due to defective LDL receptor. So those are the two main points from this discussion. Now let us look at what are the other molecules produced from cholesterol. I told you cholesterol is an essential molecule. Okay, without cholesterol, you would not have even developed into a complete, newborn child. Your essential development itself requires cholesterol. Embryogenesis itself needs cholesterol. So it's an extremely important molecule. So its notoriety in modern world is not due to cholesterol being bad. It is actually we being bad, because our metabolism evolved to store food when it is available. Remember the hunter-gatherer version of living, which happened for 99% of our existence on Earth in terms of evolutionary time. We did not have guaranteed 3 meals sitting on an armchair, okay, or couch potato, sitting on the couch and watching TV, and the only exercise being playing with the remote control, TV remote control. So they were actually running, jumping, climbing trees, and getting low calorie fruits and vegetables and tubers and so on. Occasionally an animal kill some protein from it. They didn't farm lentils and rice and wheat and crystallized sugar and so on. So therefore, whenever meal was plenty, the metabolism learned to store it. Okay, and when you weren't finding any food, or you are crossing a patch of dry land with nothing, then it used the stored food, and as a result everything was totally fine, and there are no issues. But you fast forward to 21st century, what you are having is 3 guaranteed meals every day, apart from snacks that some people munch uncontrollably. You can see some people incessantly eat snacks, and all of that are calorie. People hardly eat non-calorie food. So they are all being stored in the adipose tissue. And on top of it, there is no utilization of the stored fat, because they hardly get out of the couch. And that is why you have all the problems that we have now. So the molecules have nothing to do with it. Okay, so if you have a lot of physical work, no amount of cholesterol is going to harm you. It will all get used up. You saw how the regulation works. So anyway, now we move on to other molecules produced from cholesterol, as well as molecules coming from the intermediates of cholesterol biosynthetic pathway. So that's what we are going to look at it. A famous molecule is shown here, progesterone. So this progesterone is what regulates the estrous cycle in female reproductive system. So this is a derivative from cholesterol. So we will see in some detail how this is made. So cholesterol, an important intermediate pregnenolone. This is the one from which the steroid hormones are produced, and the pregnenolone to progesterone, and from progesterone you get in adrenal gland, in adrenal cortex, you have two hormones, glucocorticoids, which regulate glucose level and suppresses immune system, and that is why excessive use of steroid drugs will end up having immune suppression problems. And this is the reason also why steroid drugs are given to control allergy and inflammation. Okay, so naturally in our system these cortisols do that, the glucocorticoid versions. So the name itself tells you what is this. Gluco means it senses and regulates glucose metabolism. Cortico meaning it is produced by adrenal cortex. Oid refers to steroid. Okay, so it's a steroid produced by adrenal cortex, functions in regulating carbohydrate metabolism. Okay, so that's the name. That's what you get from the name and this name. And another adrenal cortex steroid hormone is this corticosterone version, mineralocorticoids. So these mineralocorticoids produce, one of the important mineralocorticoid is aldosterone. What it does is it regulates the uptake of the salts in kidney, reabsorption, in kidney. So therefore this hormone regulates electrolyte balance, which is again very critical. Okay, so these are important hormones. And you need to remember the amount of cholesterol needed to produce these steroid hormones is extremely low, because these hormones act at extremely low concentrations. So the cholesterol flowing through this pathway is really negligible compared to the cholesterol that goes to membrane or in bile acids and bile salt production. And the second set of hormones, okay, this is the adrenal hormone. I'm calling these two together as one. And the second one are the hormones produced by the gonads. Okay, in the first intermediate is testosterone, and that is converted in estradiol in ovary. And testosterone in males and estradiol in females act as the sexual hormones, and these influence the secondary sexual characteristics. And also they are also, along with progesterone, is involved in regulation of female reproductive cycle. So therefore the steroid biosynthesis comes from, so steroid hormone biosynthesis are derived from cholesterol as the precursor. And how are they produced from cholesterol? That we see in the next slide. Oops. Yeah, here. So essentially this side chain is removed, and that is removed by an interesting biochemistry. Okay, so two adjacent carbon, this 20 and 22, get oxygenated, okay, by enzymes called mixed function oxidases. And these are cytochrome P450 dependent enzymes. And we need not go into the details of these, electron transfer proteins. So you forget about it. All you need to remember is mixed function oxidases are involved in oxygenating adjacent carbons. Okay, and you get these two hydroxyl groups, and then desmolase, the next enzyme, ends up cleaving this bond, generating a carbonyl group. Okay, so this is the primary reaction involved in the formation of all the steroid hormones, the side chain cleavage. Then you have additional steps where the other carbon atoms may be oxygenated, making hydroxyl groups. So, where which hydroxyl group depends on which hormone you are making, and that detail we need not worry for this introductory class. If you remember this, that is steroid hormones are formed by side chain cleavage, and how the side chain cleavage happens, and this point that adjacent carbons are oxygenated, that is they add an oxygen to make a hydroxyl group by a class of enzymes called mixed function oxidases, and such enzymes are involved in later oxygenation steps as well in the pregnenolone precursor for the other hormones. Okay, so this is starting from cholesterol. Now I told you the intermediates in the cholesterol biosynthetic pathway also serve as precursors to make other molecules, and that is in this slide. Okay, this whole lot of molecules are made depending on what kind of organism you are. Like for example, this isopentenyl pyrophosphate, this is the active isoprene group I told you right yesterday, and from that molecule you make all of this. Let us start somewhere familiar. We'll start here. So the isoprene themselves are active molecules. These quinones that we saw, ubiquinone, and in plants plastoquinone, and these are electron carrier proteins, they are synthesized from this. Dolichols, we saw that they carry carbohydrates for making lipopolysaccharides on the membrane. So they anchor on the membrane, help in transfer of groups from one molecule to the other on the membranes. So we saw that. And phytol chain in chlorophyll. So we saw phytol in chlorophyll. So there that biosynthesis, all long chains where you have isoprene, the 2-methyl butadiene portion. So that is this portion is repetitively involved in biosynthesis of those. Rubber and cholesterol itself. Then we learned from cholesterol steroids, bile acids, and vitamin D. So this we have already learned. Then vitamin A, that again has a long chain. Vitamin E related to dolichols, that it's an antioxidant on the membrane. Vitamin K involved in blood clotting. Carotenoids, we saw, the accessory pigments in chloroplasts in the light harvesting antenna. Then these we haven't heard of, abscisic acid, gibberellic acid. These are plant growth hormones. So abscisic acid is involved in leaf senescence and falling, in the seasons where leaf fall happens, and fruits ripening and falling, and for the separation of that part, leaf for the fruit, that is promoted by abscisic acid. Gibberellic acid is involved in germination of seeds. So many molecules are produced from this isopentenyl pyrophosphate, and they all together called isoprenoids and isoprenoid biosynthesis. Okay, so these are the variety of molecules that are produced from the isoprene that is produced as part of cholesterol biosynthesis. And many fragrances, as I told you, are derived from this. Okay, so the last point in our metabolism discussion is this covalent modification of proteins we revisit. So you're very familiar with phosphorylation and dephosphorylation as a covalent modification. You have also seen here and there AMP being attached to an enzyme, and that becomes an active. Or you have also seen, what else you have seen on the way? I'm trying to recollect. Primarily phosphorylation, dephosphorylation. And we have seen this UDP, CDP attachments in activating, but I didn't discuss that in detail. So anyway, for sure you remember the phosphorylation, dephosphorylation being a regulatory covalent modification to enzymes. Similarly, there are other groups that can be added. One of them is isoprene group addition, okay, to proteins, covalent attachment, and that is called prenylation. And this can be sometimes a farnesyl group attachment, so that is called prenylation as well. If you want to be very specific, farnesylation. And sometimes it is geranyl group attachment. So, geranyl is IPP DPP joining is geranyl group, and then you have another IPP that became farnesyl. But instead, if you have two geranyl group joining, that gives you this 20 carbon geranyl group. So these are hydrophobic molecules, okay? So they help in anchoring some of the proteins to cell membranes, and that often is important for regulating their biological activity. So the membrane bound form will have an activity, or sometimes it is sequestered on the membrane and taken away from activity. And those sort of regulation is done by covalent addition of these prenyl groups. So that is prenylation. So now you will not be able to think cholesterol in a bad way, okay, after hearing all of this.